A pós-graduação está aqui, vai ser gravado? Carol, tá- pode eu que inicio a gravação? Não, tá bom. Então,
Ah,
vamos
ok.
dar início ao exame de qualificação do aluno Ian Nunes de Valença. Eu tenho um orgulho de apresentar a banca, o professor Darlan Silva Cândido, que é formado na Universidade Federal do Ceará, tem mestrado na USP em alergia e imunopatologia, tem doutorado na Universidade de Oxford em epidemiologia genômica de arbovirose SARS-CoV. Ele é ligado ao CAD, que é um programa grande de cooperação entre o Brasil e o Reino Unido, que é um centro para descoberta, diagnóstico e epidemiologia das arboviroses, e atualmente é professor associado em pesquisa da Imperial College London, onde ele está neste momento. E o professor e o doutor Anderson Fernandes de Brito, que é graduação em Ciências Biológicas da Universidade Federal da Universidade de Brasília, um mestrado pela USP, um doutorado na Imperial College, então nós temos um grande contingente de Imperial College aqui, né? Um pós-doutorado na Universidade de Yale e atualmente é pesquisador do Instituto Todos pela Saúde, na área de genômica e avaliação de transmissão de vírus. Você tem uma banca super especializada aí, Ian. Então, como vai funcionar para... O Ian faz uma apresentação relativamente breve, aberta ao público, então todos que quiserem estão no YouTube, né? E depois a arguição ela não é pública, ela é entre a banca e o aluno. Então, por favor, Ian, fique à vontade, estamos prestando atenção.
Vou compartilhar minha tela, só um minuto. Deixa eu compartilhar tudo. Todos conseguem ver?
Sim.
Certo. Bom, primeiramente eu queria agradecer, né, essa oportunidade de estar discutindo o meu projeto com todos vocês. Também é uma honra para mim ter esse tipo de discussão, especialmente com pessoas que eu considero que são realmente da minha área, né, também, e a qualificação proporciona nesse momento. I will try to start in English, but maybe there's a possibility that I will change it back to Portuguese. So my project is entitled Genomic Surveillance of Emerging Viral Threats in the Human Population from undiagnosed clinical case in the São Paulo region. So it's a project that's been done with in collaboration with many great institutes under the mentorship of Professor Nuno Faria. And in this context of emerging viral threats, we now know that due to climate change and land usage, there's the virus zoonotic spillover are expected to increase in the future. So in this work by Carlson, they show that virus sharing is correlated with population density, and it shows that in some areas of the world, it will people will have more contact with some with some types of animals, and this will increase the risks for emerging of virus. And another study also shows that and it also correlates the regions of high biodiversity with increased chances for zoonotic virus to do the spillover to humans. And here they show that South America is also a great, it's considered a hotspot for this kind of emergence. And they also show that especially for mammals and bats, the regions that have this kind of animals have increased chance for this kind of spillover. In another study, also they show that they get data from the 80s to around 2010, and they show that most of the outbreaks that we had during these years, they were a significant part were caused by viruses and also by bacteria, but they are mostly zoonoses, zoonotic virus, and they are mainly non-vector borne diseases. So we had some really interesting cases of viral emergency in the past years, and Zika, especially for Brazil, was one of these cases. And to show that it's important to have genomic surveillance, the first case confirmed in Brazil for Zika was in May 2015. And in this work, doing the sequencing of some cases of Zika, they could estimate that the real origin, time origin for the Zika was actually 1 year before, in February 2014. So we had this whole 1 year of the virus circulating in the country without anyone noticing or detecting, because we did not have the genomic capacity for that. So with the COVID, we were better prepared for doing sequencing, but still we had several deaths, especially in Brazil as well. But we also could demonstrate some of the importance of doing genomic surveillance, identifying one of the most important lineages of COVID that circulated in the world, in the Amazon uh, and the Amazon region. Also doing genomic surveillance, and the importance of this is that although it was uh, a really sad pandemic, it also prepared most of the countries, not most of the countries, but in some of the countries, to have some better genomic capacity by having more sequencing resources, such as the platforms to sequence. So today we are better prepared in this sense, as we have more uh, platforms to sequence. But we still we know that there's a disparity of the distribution of this kind of sequencing. Mm, but we have the resources to use and to do genomic surveillance. Oh, sorry. So the World Health Organization say that we should focus on more cost-benefit strategies to identify new viruses. And one of these strategies is to focus in surveillance that uh, tries to identify the virus before it gets adapted to the human population. So it's in a uh, part called emergence. So it's considered an early detection, syndromic surveillance, and we try to identify the virus when it's first doing the first jumps to the human population. And we usually only try to sequence when the virus is already spreading uh, in the whole population, which is not very efficient. Considering all this, one interesting spot here in Brazil, which is considered a hotspot for emerging viruses, is the Hospital das Clínicas, that is the biggest medical center in Latin America. and it has more than 9,000 health workers, not only health workers, workers in general, and it receives more than 250,000 patients per month. Sorry. And once we can have the genomic data that we can apply in this kind of settings, having the public health questions, we can answer them by combining some epidemiology and phylogenies with the sequence data, and then we can transfer this information to actionable information for the public health sectors, such as knowing the introductions, and if we're having community transmission, the time and location of the outbreaks, and also identifying clusters of transmission, doing the epidemiological parameters, such as the Zika case where we could identify the first detection, and also clinical parameters that can be inferred from this combination of data. So the possibilities of having genomic data are really uh, uh, great, and we can use that in our favor. So the main strategy to do the genomic surveillance is using the next generation sequencing platforms, which uh, produce a large amount of data from virtually any kind of sample. And we have mainly two types of approaches for NGS, which is target sequencing, where you use um, specific primers to sequence a region or an organism. And also, we have the untargeted sequencing. And here we have the metagenomics approach we use in this study. And it's considered an unbiased approach because we don't need to have an information prior to sequence the sample to know and to extract information from the sample. So a standard approach for doing sequencing is once we have the sample, we extract the genetic material, do the cDNA synthesis, and then we can do either target primers or random primers. In our case, we do the random primers approach. And the main, this is one of the simplified uh, example of how the sequencer works. So the sequencing will include the platform, either Illumina or Nanopore or Pacific Biosciences. We read the raw data and transfer, convert it to the DNA information. And here we're gonna usually have, especially for a clinical application, removal of the host and the filtering. And we're also gonna do the multiplexing, which is referred to the barcode for the identification of the samples. And then we do the one of the most important parts for viral identification, which is the metagenomic classification that implicates the taxonomic labeling of the reads that we sequenced. And considering that the Oxford nanopore technologies are um, especially good for a clinical environment. They can do real-time sequencing. They can produce longer reads that also sometimes helps to recover the viral genomes during the assembly. And they also implement a lot of interesting technologies, which is the case of the adaptive sampling. That is a technique aiming to enrich or deplete a specific a specific sequence or region that we want. So in our case, as we are interested to deplete the host DNA from our samples, during the sequencing, for each flow cell that we have the pores, as is the how the nanopore works. So each flow cell has many pores like this in the flow cell, and it reads the DNA strand when it passes through the pores, and it changes the electric voltage of the pore, and it can be read from a sensor in the flow cell. The adaptive sampling works by once a part of the read is passed through the pore and it's read on the sequencing, they will be mapped against the reference that you chose. And if you don't want the sequence to be continued to be sequenced, the pore will revert the voltage, and the read will be uh, ejected from the pore, and other reads can be used in this pore. So in the sense that we don't sequence human reads, we are enriching our samples from other material that are in the sample. So it's an indirect way of enriching the viral and bacteria reads that we have in the sample by not sequencing the host DNA, or at least trying to reduce this. Well, this is what the theoretical uh, uh, objective of the adaptive sampling is. So this would be the perfect outcome of using adaptive sampling. But we saw that in practice, it's not so simple. And after having the data from the sequencing and doing all the filtering steps and quality controls, we're gonna end up with the metagenome classification. And Kraken 2 is one of the gold standards for this type of classification. And it mainly works by trying to classify each read that we sequence to a lowest common ancestor. So it's very efficient and very fast the way it classifies compared with others aligners and classifiers such as BLAST, which is very sensitive, but it takes too long to classify all reads. And it has a database that contains all the chimeras. So it works by this dividing all the genomes in chimeras, which is generally 31 mer size. And it has this pre-built database, and for each read that you have, it's gonna try to classify the reads by summing all the reads that classified for each node in a taxonomy tree that it has on the database that refers to the taxonomy level of classification, and you're gonna label your read to the lowest taxa that it can find by summing up all the weight in the nodes. Sequences that don't have any chimeras in the database will be left unclassified by Kraken. So that's a simplification of how the reads are being classified. So our project aims in, this would be an overview of the project. So we try to focus on undiagnosed samples, in this case in clinical settings from health workers, trying to maximize our chance of identifying different viruses. So ideally would have the patients that are symptomatic, and we would collect the sample in the Hospital das Clínicas, and then we would submit the sequence to the nanopore sequencing, do the main steps of bioinformatic analysis, which is pre-processing, dehosting, the taxonomy classification assembly. And this would be ideally if we had a platform to sequence all the samples and that we wanted to give feedback to the doctors. So as this quick report could take 3 days or 2 days to be made, and after that we also would do the more in-depth analysis, such as the phylogenetic analysis. So the objectives of my thesis is first to standardize clinical metagenomics for detecting emerging viral threats, to do the metagenomics diagnostics of undiagnosed case using nanopore sequencing, and to enhance sequencing accuracy in viral classification with advanced bioinformatics. So the overview of my project is that we had around 2,091 samples from nasopharyngeal swabs, and we filtered this by only getting the samples that were negative for SARS-CoV. So this includes all these 2091 samples. And then we selected only for patients that had experienced fever, febrile illness. And then we ran a respiratory viral panel that included 4 main respiratory virus that circulates in Brazil, which is RSV, syncytial virus, rhinovirus, influenza A, influenza B. And then we separated the samples that were negative for the 4 virus and the ones that were not. And here I show that 110 were positives for at least 1 virus, but we did not sequence all of them yet. And also because we were, in the beginning, we were not interested in this kind of respiratory virus, but then we saw that it would be interesting to also sequence and see it would be used to standardize the metagenome protocol with the CT values that we found. And this is the distribution of the libraries that we sequenced so far. So we had 2 libraries which we had not spiked COVID samples, but only COVID samples from other studies that we had also CT values for them. And then we did 2 libraries with same samples to compare the DNA treatment we were using with R9's flow cells. And from that, we did 2 libraries that included positive samples for this virus, the virus that we identified from the viral panel. And the rest would be the virus that were negative for all this virus and that we are sequencing. So we applied this viral respiratory panel, and we found that RSV had a really high CT value compared to RNV, to rhinovirus, which we could identify more. And we only could identify 1 influenza through metagenomics, but we had more samples that identified for influenza in the viral panel. And we also could not identify any influenza B in this run that we did, that we applied the respiratory viral panel for the 200 samples. And for the sequence, we are using a protocol that were developed by Ingra. It's called the Smart 9N, which uses random primers to do the sequencing. So it theoretically can sequence all the genetic information in our samples. And it's also a relatively fast protocol. It takes about 6 hours to be completed. Of course, there is some variations, depends on the amount of samples we are sequencing, but it's more or less the time that it takes, and it also costs. It's a considered low cost sequencing protocol. So that's why we choose, because if we want to do genomic surveillance, we want to do as cost efficient as possible. And it costs around $30 per sample to be sequenced. And also this varies depending on how many samples you have per library. And for the bioinformatic workflow, we have this standard, it could be considered standard workflow, and it changes sometimes because we need to be improving it when some software is released, such as happening with the pre-processing, with the release of other base callers. So Doutorado is a base calling process. So the pre-process step starts with the base calling during the sequencing. And for our samples, we applied adaptive sampling for almost all of them. It's an optional step. And after the QC analysis, we also do the dehosting in the bioinformatic process. So we map the reads against the human genome reference with Minimap. And the remaining reads will be classified with Kraken using the standard database, which includes fungi, viral reads, bacteria, human reads or human genome, and eukaryotes as well, other eukaryotes, such as archaea, oh no, not archaea, such as, sorry, I just forgot the name of the other cards, but includes other types of species as well. And then we take the classified reads from Kraken and do the assembly with Fly, which is more adapted to nanopore sequences. And then we run BLAST with this contigs that now we have less data, we can do the BLAST and then confirm the results that we had with Kraken. So these unclassified reads are the reads that were not labeled, any taxonomic label. So they are particularly interesting if we're trying to find a really divergent virus, which that's rare to happen, but it could happen. And then we can do the phylogenetic analysis of the consensus or contigs that we found. For this preliminary data, we used mostly NextStrain to do the phylogenetic analysis. That is an analytic and visualization tool and can do real-time tracking of pathogen evolution because it has a huge database online that we can compare our sequences to. And so it improves our epidemiological understanding and improve the outbreak response in real time. So in this presentation, I divide my work in 3 chapters, considering my objectives. So this first, and I'm gonna present some of the conclusions that we had for each chapter, although it's ongoing work and some of the data is still not complete. So this is a summarizing of the data, the sequencing data we found, considering the viral panel and the sequencing reads. So our total N of samples with combining these two reads that we had some positive samples for the virus and metagenomic results are 45 samples. And this N will increase in the future. But so here we have the median for each variable. and also the interquartile range. And we see that there's many variation, a lot of variation for either for the viruses as for the variables that we tested. So for each sample, we see that the amount of total reads varies drastically, and for each virus. And we see also that for SARS-CoV, we had a better quality of sequencing because it was specifically a library that we did not use the adaptive sampling, which was not expected. One of the results that we could find is that we found a limit of detection uh, where we saw that after a CT of 30, we could not identify any of the virus that we had in the viral panel. So this result also compared with some of all the results that we had before. And for nanopore sequencing, at least, it seems like CTs above 30, it really difficults the detection by metagenomics. And we see that as the CT values increases, the viral load also, as the CT values increases, the viral load decreases, and it gets harder and harder to identify the virus. And that's one of the reasons we could not identify any RSV. All of them were with CTs above 35 or 34, and through metagenomics, there was no possible detection for RSV reads. So one of the comparisons we did was with these two libraries, the library 1 and 2, where in both we used adaptive sampling, but we compared how the DNA treatment would affect the amount of reads, the viral reads, and also the bacteria that are DNA, but this data are not included here. So for these libraries, we had 8 samples that were from our nasopharyngeal swabs study. So these are the same samples. And we had 3 samples here from barcode 9 to 11 that were SARS-CoV samples with known CTs. So we could test if the library was like successful or not. And we had 1 negative control here too, as we have in all the libraries we sequence. And we could see that the DNA treatment, as expected, um, it increases the proportion of our reads significantly, and it also decreases the percentage of human reads, as we are also expecting, and the number of total reads increases. The throughput, it increases compared with the library that we're using also adaptive sampling, but no DNA treatment. In one of our samples, we identified influenza in barcode 28 from the library 4 that had that was a test including samples positive for the viral, for some virus from the viral panel. And it's an example of, although the sequence had a really low coverage, which is questionable, but we could at least say it was H3N2 strain, and we could also identify the clade and short clade using the next strain platform for influenza. We did the same with the SARS-CoV-2 sequence that we could retrieve. So this sequence were of really high quality. They are mainly from the library 5, and all of them, almost had more than 99% coverage and had a really high depth. So it shows that we can achieve a really high quality of data genome if we have the right sample conditions. And as expected, they were all Omicron, as this data, I think I did not mention the data collection, right? But it was all from the after July of 2022 until December. So we had mainly Omicron in the country already. So for this chapter, we see the conclusions were that CTs above 30 seems to limit the detection through a metagenome protocol. So higher CTs are inversely correlated with viral loads, the viral is detected. And DNA treatment significantly improves the number of viral reads sequenced. And with the right sample conditions, we were able to generate high quality genomes. So for the second part, we did try to see the metagenome diagnosis of undiagnosed case using nanopore sequencing. And in library 3, we first did the sequence without knowing if they were, without using the viral panel. And we are now identifying the virus, running the viral panel in these samples. But here we could see already some of the, identify some viruses that were not in the viral panel, such as the human metapneumovirus and one strain of coronavirus, the human coronavirus, the 229E. That we cannot retrieve the complete genome yet, but the analyses are still ongoing. We can maybe able to retrieve some of the genome to do some of the phylogenetic analysis. But for this library, we started some microbiome analysis using the bacteria reads that we identified. So for these samples, we normalized the reads per 100,000. And we need to do more analysis on that, but I can show what we can also do for the undiagnosed samples that we have. And here, at least, we can see that most of these bacteria are commensal or in the environment, these three at least. But we see that we can have maybe some interesting results here with the Klebsiella pneumoniae that's common in hospitals settings, clinical settings. And one example of classification for undiagnosed cases using nanopore, and that it came recently also by Ingra, it's the two cases that were suspected to be yellow fever, fatal cases, patients that were fatal cases, and they were suspected to have severe yellow fever. After metagenomic analysis, they could identify the strains for the Brazilian virus, also known as Sabia, and it changed the diagnostic of these patients. We are also doing the microbiome analysis for the samples that we could not identify the causes, because just some of the 2 cases from the 8 patients were Sabia virus, so the rest still unknown. But maybe there's some agents that we can discover from the microbiome analysis that we can do also with the metagenomics. So the conclusion for this chapter so far is that the detection of unknown pathogens is viable through untargeted metagenomics with nanopore. That microbiome analysis can help the interpretation of undiagnosed samples. And from my project, 8 more libraries are to be analyzed, combining viral and bacterial data. So far we had only 7, and now we had more 8 to analyze. For the last chapter that we are trying to enhance sequence accuracy and viral classification with advanced bioinformatics. This is the also ongoing analysis. And one of the things that we do is to compare some of the pipelines, bioinformatic pipelines that are out there, such as Arcadia and App2Me. I forgot to put the reference for Arcadia, but App2Me is from Oxford Nanopore. And they are both standard, but they have some differences. And in our pipeline, we try to combine the goods on each one of them. And so, but we do the analysis manually. And so it's not automatic. There's no automation yet, but we are aiming in doing that as well. This is some of the results for the Arcadia pipeline. And I don't include the taxonomic classification here, but all of this is automated by the Arcadia. They are a really new pipeline, so they have still they are improving the the software, but we are using just the preliminary to see the preliminary results. So it generates some interesting statistic for the sequencing that can be generated manually, but they do all of this as automated pipeline. So they do the assembly, which the App2Me pipeline don't do, and also they analyze the quality of the assemblies and the number of contigs that we generate for each sample. And another test that we did was to improve the base calling. So today, Nanopore, that was known to have really low quality sequence accuracy. Now they are improving that only by improving their algorithms. So they use neural networks to do this, and we were comparing the super accuracy base calling with the fast base calling using Doutorado. And although this is very computationally expensive, it's worth to do the basic calling with super accuracy because we can really increase the average quality. Here in this is preliminary result as well, but we are only comparing the average read quality here. The read lengths are not scaled in a right manner, and we have even reads that achieve 25 to almost 28 and quality, and we have some other reads that are achieving 30 in the Fred score, which is compared to Illumina, although still we have some ground to go, as the average is around 16, 17 QC. And we also did some statistical analysis for the adaptive sampling that we did, and we found out that it does not improve the variance that we were expecting. In fact, it's reduce the total throughput, and we are investigating why this is happening. And this would be example metagenomic report if we were doing automated metagenomic analysis for a clinical setting. So this would include all the QC analysis and the virus that we found, and also we could try to attribute some statistic for the positive virus that we find, and to include if we can generate or not the consensus of the virus, and also the cope this with the microbiome analysis. Oh, sorry. So the conclusions for chapter 3 is that more accurate base calling improve the quality of reads and can reduce the amount of false positives. Rebase calling the raw reads after sequencing can be worth to extract the maximum information of the data. It's possible that adaptive sampling is not working expected due to computational constraints. And with our current tests, adaptive sampling is not enriching viral reads. So some of the considerations is that for the metagenomic classification is that classification sensitivity and specificity highly depend on how complete and accurate the user reference database is. So this is could be considered a limitation of the study, but it's to all metagenomic studies. So because the entire microbial biosphere is still far from being completely sequenced, so we have some missing partial genomes that can introduce bias and reduce this classification performance. And because of this, some reads can be misleading, as they are wrongly classified to other taxons. And we have this problem of some genomes that are submitted to the public database. Sometimes they have parts of other genomes inserted, so it ends up Kraken ends up classifying these regions, and so it attributes wrong taxonomic labels. So this is considered a problem of contamination of the database. But all of this can be fixed. So my next steps are analyzing the remaining samples in libraries. They are in total 8 now, so far. Do the phylogenetic analysis for all the virus that we found. Do in-depth analysis of the bacteria data that we are including in this study. Implementation of Kraken unique that aims to reduce false positive. Including the diamond, which is an amino acid-based qualification results that can improve the sensitivity, and doing that with Kraken. Adding the false positives metrics, which can be important to clinical for a clinical setting and a clinical report. Trying to investigate contamination either with virus and bacteria and other kind of organisms as well. We need to add the clinical metadata. We did not receive it yet from the hospital, but we are expecting to receive soon. And also to include all the metagenomic runs that we had in our institute, so we can have a significant amount of data to do a machine learning approach to identify optimal methodology to apply for the next runs in a context-specific manner. And I think that's all. Thank you so much, everyone. I almost extrapolated the time.
So we finish here the public